Welcome viewers. Welcome to the session on culture medium and the types of culture medium. What is a culture medium? Culture medium performs an important process in microbiological studies. It is generally composed of inorganic and organic nutrients mixed in a defined volume of water adjusted to a suitable pH required for the growth and development of microbes in laboratory. Now, what kind of culture medium depends upon the nutritional requirements of the bacteria. Whether we want to grow autotrophs or heterotrophs, the composition or the type of culture medium is designed accordingly. Microbes that grow in or on the culture medium are known as microbial cultures. Most of the microbes are able to grow on this synthetic medium, but however, there are certain group of microbes which fail to grow in lab conditions. Now let us move to the types of culture medium. The type or the classification of culture medium can be done in various ways. The first system or the type of classification is based on its consistency. What is consistency? That is the physical appearance of culture medium. The second way in which we can classify the culture medium is on the basis of its nutritional components. Means what kind of nutrients have been added to the medium. The third system or type of classification of culture medium is based on the functional use. Let us come to the first system of classification of culture medium, that is consistency, which means the physical appearance of the culture medium. If the culture medium is liquid in nature, then it is known as broth. The liquid culture is done when we require a large number of population of bacteria in a small defined period of time. Then we go for liquid culture. The solid medium is done by addition of agar. Agar generally is added at a concentration of 1.5 to 2% to the culture medium. It is then sterilized by means of autoclave. If the solid medium is dispensed into culture tubes and hardened in a slanted position, then it is known as agar slants. The slants are generally used for maintenance of pure culture and for biochemical studies of that particular microbe. Whereas the second form of semi-solid medium is agar stab or agar deep. What is agar deep? Agar, when solidified in an upright position in the culture tube, it gives rise to agar deep. It is generally used for the growth and cultivation of anaerobic bacteria. When the sterilized culture medium containing agar is spread into a petri plate, it covers a large surface area and it is known as agar plate. Agar plate is generally used for the isolation of pure culture from a mixed group of bacterial culture. So this was on the basis of consistency. The other two types of media which can be classified or which can be grouped under this category are semi-solid medium and biphasic medium. Semi-solid medium generally contains low amount of agar. As the name suggests, the medium has a semi-solid consistency. It is formed by the addition of agar of 0.5 to 0.7%. This kind of media finds usage in studying the bacterial motility. As we know that there are certain group of bacteria which moves with the help of flagella. This kind of medium can be used to study the bacterial flagellal movements. Like for example, certain transport medium like Stewart and Amis medium also are semi-solid in nature. Now the last group which belongs to this category is biphasic medium. Biphasic medium, as the name suggests, consists of two phases. One is the solid phase and the liquid phase.
Ma'am, what is the function of biphasic media?
The function of biphasic medium is that we do not need to open the bottle for frequent subculturing. Now, since it consists of two phases, solid and liquid phases, so the bacteria is inoculated into the liquid phase. After it sustains or shows growth in the liquid culture, it can be tilted over the solid medium which is present in the same bottle, so allowing the subculturing of bacteria on a solid phase. So this prevents the requirement of opening the mouth of the bottle for frequent subculturing, which can lead to the contamination of the culture. Now let us move to the second system of classification of culture medium, that is on the basis of its nutrients present in the culture medium. We can classify the culture medium on the basis of its nutritional components into mainly three types. One is semi-synthetic, synthetic or defined, and complex medium. Now first let us move from synthetic to complex. What is synthetic medium or defined medium? Synthetic medium or defined medium means they contain ingredients which are known both qualitatively and quantitatively. For example, if we look at the culture medium of BG11, which is often used for the growth and cultivation of algae in lab, if we look at the composition of BG11, it will show that it is a mixture of simple inorganic salts and carbon is present in the form of carbon dioxide. So this kind of medium, synthetic or defined, can only be used for those group of organisms which have the ability to synthesize the macromolecules from this simple inorganic compounds. We can divide further the microbes on the basis of its requirements into two types. The BG11 or any synthetic medium generally grows those kind of microbes which have specific growth requirement. Like how they have the specific growth requirement, because if we want to study what is the kind of growth requirement needed for the growth and cultivation of microbes, then we go for synthetic or defined medium. While those group of organisms which can be grown on a wide variety of nutrients, or they can be grown on medium containing extracts from different plant and animal sources, that it is in the complex media, belong to the non-fastidious group of bacteria. While the fastidious group of bacteria, which is more specific in their growth requirements, are grown in enriched medium. The second medium which falls under this classification is complex medium. Complex means, as the name suggests, the ingredients are not known precisely. Like these contain extracts from different plants and animal sources of undefined composition. Mostly the complex medium contains peptone, beef extract and other things. Peptones, which is derived from the incomplete proteolytic digestion of casein, soya meal and other proteins.
Ma'am, what is the function of yeast extract in medium?
The function of yeast extract is it is derived from brewer's yeast and it is a rich source of vitamin B, carbohydrates and proteins in the medium. Now the microbes which are growing in medium containing yeast extract are able to break down the yeast extract and they can degrade the yeast extract and produce their necessary macromolecules. Now similarly we have beef extract. Like beef extract derived from animal source is a rich source of organic carbon and energy, because we know that organic carbon is oxidized to obtain energy through the process of respiration. So examples of complex medium are tryptine soya broth, McConkey agar. Why? Because all these kind of medium contain ingredients like tryptone, peptone, etc., which are again derivatives of plant and animal sources, and we do not know the exact components of beef extract. Now let us move to the third part of your medium under this classification, that is the semi-synthetic medium. Semi-synthetic medium is generally a mixture of the synthetic compounds and your complex compounds. Like for example the nutrient agar, which we use in our lab. It's generally a mixture of peptone, beef extract and NaCl. So here you know the composition or amount of NaCl. But what is the component or what is the composition, exact composition of beef extract and peptone is not known. So it's a mixture. Other examples of semi-synthetic medium are potato dextrose agar medium, which is again used for the cultivation of fungus in lab. The third system of classification is classification of culture medium on the basis of its functional use. That is, how can we use the culture medium for our various microbiological function, or how we can use culture medium to differentiate between the different group of microbes growing on the same culture medium. One type is basal medium.
Ma'am, what is basal medium?
Basal medium are relatively simpler medium. Why simpler? Because they contain nutrients which are providing a base for the synthesis of other macromolecules of bacterial cell. Like an example of basal medium is nutrient broth or nutrient agar. Nutrient broth or agar contains ingredients like peptone, beef extract, NaCl, and they can be used for the growth of non-fastidious group of organisms. Since they contain ingredients which are non-specific in nature, therefore they are mainly used to grow a wide variety of bacteria in lab. The other examples of basal medium is peptone water. Peptone water medium is a type of carbohydrate fermentation medium, and it is used to study water microbes in it. It contains peptone, NaCl mixed in a defined volume of water and adjusted to pH 7. Now let us move to the second group of media which falls under this category of media of functional use, that is minimal media. As the name suggests, it contains the minimum ingredients which is required to support the growth of bacteria. These kind of medium are generally used to select the recombinant or the wild-type bacteria. Why? Because these kind of media generally lack the presence of one or two specific amino acids. So they can be used to grow those kind of bacteria only which can synthesize the absent amino acids. So this helps us to study the recombinant or the recombinations which occurs in nature. The third kind of media which falls under the classification of media based on functional use is selective media. As the name suggests, this kind of media can be used to grow only a specific group of microbes. Why? Because these kind of media contains antibiotics or bile salts or certain dyes which prevents the growth of a specific group of bacteria while permitting the growth of a particular group of bacteria. Examples of selective media are EMB, commonly known as eosine methylene blue. How does it select? Eosine methylene blue, it contains dyes like eosine and methylene blue which retards the growth of gram-positive bacteria. It only allows the gram-negative bacteria to grow. Example is E. coli, Enterobacter aerogenes, which proliferates profusely in this kind of medium. The other kind of selective medium are McConkey agar. McConkey agar is again useful for selecting the lactose fermenters or the enterobacteria. McConkey agar generally contains bile salts which prevents the growth of gram-positive bacteria. Similarly, we have mannitol salt agar which contains high concentration of salt of around 7.5%, which prevents the growth of a wide variety of bacteria. This type of medium is very specific and it is used for the growth of a specific group of bacteria. Medium like EMB, that is the eosine methylene blue, McConkey agar are generally used to study the water microbiology. Why? Because generally the contaminated water contains E. coli which are generally passed from the human intestinal tract to the water supplies. Now, if there is a water contamination with E. coli, then it will be able to grow on EMB plates or on McConkey agar plates, and hence it is useful to study whether the bacteria is present in the supplied water specimen.
Ma'am, how can we isolate cellulose degrading bacteria from soil sample?
The soil contains a mixed population of bacteria. We can isolate the cellulose degrading bacteria from soil by selecting them on a medium containing only cellulose. So those group of microorganisms which are able to degrade cellulose as their organic carbon source or energy source will only be able to grow, while the other groups of bacteria which requires other carbon sources such as sucrose or glucose will fail to grow in this kind of medium.
Ma'am, if we have a mixed sample of microbes like fungi and bacteria, how can we differentiate them?
They can be differentiated on a selective medium. Generally, we know that the fungus or yeast, they prefer a low pH. So if we supply the mixed bacterial culture and yeast culture on a selective medium containing low pH, then only the yeast will grow or the fungus will grow and the bacteria will fail to grow. In this way, we can select the yeast from bacteria, and if we want to select bacteria from yeast, we can grow them in a media containing neutral pH. This will allow only the bacterial cells to proliferate and grow while eliminating the yeast and fungal cells. The most important group of another kind of media which falls under this third system of classification is differential medium. Differential medium, as the name suggests, it helps us to differentiate between two closely related species of bacteria or two different closely related strains of bacteria on the same culture medium. How are they differentiated? They are differentiated with the help of a certain physiological function or a presence of a certain biochemical function. Let us look at the examples. The medium which we studied in the selective mode can also be used for differentiation once. How? Like the EMB plate. EMB, which stands for eosine methylene blue agar plates, can be used to select between lactose and sucrose fermenters. Lactose are generally the Enterobacter, they perform lactose fermentation, and they prefer a medium composed of lactose. Now generally, the E. coli will grow on EMB plate producing a green metallic sheen, while Enterobacter aerogenes will grow in as a pink to red colonies and will not produce a green metallic sheen, while the other group of bacteria like Proteus vulgaris and Salmonella typhimurium will grow in this kind of media without producing any characteristic properties. So in this way, we can differentiate between E. coli and Enterobacter from a mixed sample. Similarly, the other examples of differentiating medium like the McConkey agar is another example. McConkey agar contains the indicator neutral red. This neutral red shows changes in color due to pH changes. It is used to differentiate between the lactose and non-lactose fermenters. For example, E. coli and Enterobacter will ferment lactose and produce acid as a byproduct of fermentation. The acid, which will cause the change in pH of the medium or will decrease the pH of the medium, will cause a change in the indicator of neutral red. How? The neutral red becomes deep pink in color in presence of acid. Thus, E. coli will produce deep pink colonies, while Enterobacter aerogenes will produce deep pink mucoid colonies. The nature of colonies changes from E. coli to Enterobacter, while the other group of bacteria like Proteus vulgaris and Salmonella typhimurium will simply grow on this medium without producing any color change. Why? Because they are unable to utilize the lactose. Similarly, we have mannitol salt agar, which is used to select the mannitol fermenters. Mannitol, which is used as a source of carbohydrate for Staphylococcus aureus, is used to differentiate on these kind of medium. Mannitol salt agar contains the indicator phenol red. Phenol red changes in color due to pH. Like when mannitol is utilized by this group of bacteria, that is Staphylococcus aureus, it will produce a color change. The color change is from pink to yellow. Why? Because the phenol red changes color in presence of low pH. While the other species of Staphylococcus, that is Staphylococcus epidermis, will produce no color change as it is unable to utilize mannitol and thus unable to do fermentation process, and is not producing acid, which will produce the change in the color of the indicator. The other group of differential medium are blood agar. Blood agar means the blood is added to the medium to make it more specific. These group of medium can be used to differentiate between hemolytic and non-hemolytic group of bacteria. Hemolytic group of bacteria are only able to grow with the help of your nutrients supplied from the red blood cells. According to the type of hemolysis, the group of bacteria can be differentiated into alpha hemolysis, beta hemolysis and gamma hemolysis. Generally, the strains of streptococci are grown in this differential blood agar medium. The species of streptococcus can be used to differentiate on these kind of media. The other kind of media which falls in this system of classification is enriched media. Enriched medium, as the name suggests, is nutritionally rich medium. Why? Because this nutritionally rich medium are required for the growth of fastidious group of organisms. They require nutrient-rich medium to grow. For example, there are certain ingredients like blood serum, egg yolk added to the basal medium to make them nutritionally rich. The blood serum, when added to the basal medium, forms the blood agar medium.
Ma'am, blood is a good source or a good medium for growth of fastidious organisms. Do these organisms grow inside our body?
Yes, they grow inside our body also. They produce blood-borne infections. But however, these group of organisms like Streptococcus, there are certain species of Streptococcus which causes blood-borne infections, are unable to penetrate very easily our system. Hence, they have to cross certain cellular barriers in order to enter into our reticuloendothelial system. Now, the blood agar generally provides nutrients like hemine and NAD to this group of organisms. Blood agar medium can also be used to select certain groups when antibiotics are added to them. Like we have another example of a medium, enriched medium, that is chocolate agar medium. Chocolate agar medium is produced when the red blood cells are heated and lysed. So what happens? The nutrients which are present inside the red blood cells are leached into the medium, making it more nutritionally rich, and after heating, the red blood cells loses their integrity, and when added to the medium, it forms a chocolate color. Hence, the medium is known as chocolate medium, since the color comes similar to that of chocolate. The group of organisms which is used to be grown in this chocolate agar medium is Haemophilus influenzae. Now, as I told you, if we add antibiotics to the chocolate agar medium, it makes it more selective. Like if we want to grow genus of Haemophilus only, then we can use the bacitracin, which will only permit the growth of Haemophilus on this chocolate agar medium. Otherwise, the chocolate agar medium are non-specific in nature, and it can be used for the growth of those kind of pathogens which require blood for their growth. The other kind of medium is reducing medium or anaerobic medium. As the name suggests, it is anaerobic in nature and used for the growth of specifically anaerobes. How the media is made anaerobic? It is generally with the help of addition of certain compounds like sodium thioglycolate. Sodium thioglycolate depletes the dissolved oxygen present in the medium, making it anaerobic and facilitating the growth of anaerobic bacteria. Besides this, there are certain other components added to the medium like cysteine, iron filings to keep the medium reduced. Now, whether the medium is reduced or not, there is presence of certain indicators like methylene blue. Methylene blue remains colorless when the medium is reduced. If the medium becomes oxidized, then there will be a change in the color of the medium. Similarly, there are other nutrients also added to the group of this reducing medium. Transport medium is generally used when the collection of clinical specimens, they have to be sent to the lab for diagnosis. So this clinical specimen generally contain pathogenic bacteria which need to be diagnosed that what kind of pathogenic bacteria is present in this clinical samples. So these are then transferred in a medium which prevent drying or desiccation of the pathogenic bacterium, and it prevents the contamination of the pathogenic microorganism with other kinds of microorganisms. The commonly used transport medium is sodium thioglycolate broth for anaerobic group of bacteria and Stewart's Ami medium as the collection of clinical specimen after being transported to the lab. The other kind of medium is maintenance medium. The maintenance, as the name suggests, generally is used when the microbes only need to be maintained in lab. They did not enter the growth phase, but they need only to be maintained. These kind of medium generally omit the use of glucose. Now we come to the conclusion of types of culture medium. So we can conclude that what type of culture medium we want to use in our lab depends upon the usage, and what type of bacteria we want to isolate again depends upon a purpose. If we want to isolate E. coli or we want to see water contamination is there or not with E. coli, then we will go for differential medium or selective medium. If we want to grow fastidious group of organisms, then we will provide them with enriched medium. If we want to study the anaerobic bacteria, then we can do it either in agar dips or producing the reducing medium. So the kind of medium depends entirely on our purpose of study.